Passaging, a method of speeding up the adaptation of a virus to a new host. This does not involve direct manipulation of the genome, but takes advantage of the virus's natural tendency to mutate as it replicates. Receptor binding domain, the protein of a virus that binds to a receptor on a host cell. Receptor binding motif, the portion of the receptor binding domain that merges with the receptor of the host cell, permitting the virus's genetic information to enter the cell and the virus to be replicated. Rescue, the term of art for synthesizing a virus from its genome sequence. SARS-CoV-2, the name given to the virus that causes COVID-19 by an international consortium of taxonomists. Synthetic biology, the creation of a virus or bacterium from its genome sequence. Small segments of the genome are manufactured, then inserted into a bacteria and/or yeast, whose cellular organelles put the sequences together in the right order, eventually resulting in the reproduction of a functional virus or organism. Virion, a new virus made by an infected host cell. Virology, the study of viruses. List of characters. This book describes the work of scientists who became central players in the drama surrounding the origin of SARS-CoV-2. Several were born and raised in China, where traditional naming order puts the family name first, followed by the given name. In the last 20 years, Chinese scientists have forged partnerships with Western researchers, and their work has frequently appeared in Western journals, where the traditional Chinese name order is reversed, the family name coming last instead of first. Both are included in this section. Basil Arif, Scientist Emeritus of the Laboratory for Molecular Virology at the Great Lakes Forestry Center in Sault Ste. Marie, Ontario. He is a longtime colleague of Zhihong Hu and Shi Zhongli of the Wuhan Institute of Virology, and is an associate editor of Virological Sinica, the Wuhan Institute of Virology's peer-reviewed journal. Ralph S. Barrick, a professor in the departments of epidemiology and microbiology and immunology at the University of North Carolina, Chapel Hill. He is the leading expert on coronaviruses in the U.S., and has worked with Shi Zhongli to produce chimerical coronaviruses that have pandemic potential. Chen Wei Chen, born and educated in China. She is a major general in the People's Liberation Army, its leading bioweapons and Ebola expert. She tested her vaccine for Ebola at the National Microbiology Laboratory in Winnipeg, thanks to Zhengguo Chu. She was decorated by Xi Jinping for her efforts in finding treatments for SARS-CoV-2. She created China's first SARS-CoV-2 vaccine. Cheng K.D., Kuding Cheng, Chinese-born and a Chinese, American, and Canadian-educated expert in proteomics. He is the partner of Zhengguo Chu. Both worked at the National Microbiology Laboratory in Winnipeg until they were marched out by the RCMP in July 2019. Peter Daszak, UK-born and educated parasitologist. He is president of EcoHealth Alliance, a U.S.-based charity that funded some of Shi Zhengli's work on coronaviruses via grants won from U.S. government agencies, the United States Agency for International Development, USAID, and the National Institutes of Health's National Institute of Allergy and Infectious Diseases, respectively NIH and NIAID. DRASTIC, a loosely connected group with expertise in banking, data management, journalism, anarchism, virology, and genomics. They found each other on Twitter as they each began to informally investigate the origin of SARS-CoV-2. Richard H. Ebright, molecular biologist and professor of chemistry and chemical biology at Rutgers in the U.S. He is a vocal critic of gain-of-function experiments and USAID's $200 million PREDICT program, which aimed to predict when a virus might spill over from animals into human populations and cause an outbreak of disease. Ebright was one of the first leading scientists to ask whether SARS-CoV-2 might have escaped from a lab, Shi Zhengli's lab. David Evans, professor in the